It's taken a little bit, sorry. And of course, I'm going to take you through the whole presentation before I start. First of all, I'd like to thank the organizers for inviting me. And I have to say that after Anna's talk and my talk, you're all going to be experts on soft X-ray tomography in the very least. So I'm going to start by saying that I'm Maria, I'm the principal beamline scientist of the equivalent beamline at Diamond, B24. And I'm going to talk to you about some latest developments of the beamline, especially the partnership between laser light microscopy and synchrotron radiation. So a lot of this, or some of this, will sound similar to what Anna took us through. I'll start with who we are. I don't need to introduce Diamond, the UK synchrotron, but I'll say that B24 is part of the biological cryo-imaging group, along with EBIC, which is the National Facility for Electron Microscopy. This is our website. Hang on, let me move this here. This is our website. So you can find here information about us, the method, the theory, how to approach the beamline team, and how to apply for time. And this is the lovely team that we have at B24. Moving on, what we do. This again is a little bit reminiscent of what Anna was saying. We look at the highlighted specimens for us as cells, what's happening within cells, cell populations, organelles within larger macromolecular complexes, and we can go to the other extreme for thin tissue samples. So we use both fluorescence microscopy and soft X-ray tomography to a maximum resolution of 25 nanometers, and both of these techniques are 3D, so the imaging is always 3D. In a nutshell, we're here to, the team at B24, to design and build X-ray and visible light microscopes and to develop what we like to think as user-friendly and certainly cutting-edge microscopy platforms. And we have a correlative microscopy workflow that I'm going to describe in a little bit. Two main microscopes, high-end microscopes, X-ray microscope and super-resolution fluorescence. And we're here to help from the very beginning with designing experiments all the way to sample preparation, sharing protocols, data collection, data processing, data imaging analysis, all the way to correlation. So I'll start by introducing the correlative cryo-imaging workflow at B24, which starts very much from cells, culture cells, then live cell imaging, plunge freezing, going into cryolight microscopy, super resolution and cryogenic temperature. And then we go to soft X-ray tomography, all the way to the in silico work where we generate tomograms, do the 3D correlation and the like. Different users can enter this workflow at any point based on their expertise and facilities. But I wanted to stop a little bit and sort of describe what is this sample that I'm going to be talking about. We use a flat geometry, we use TM grids, the same like Anna showed before. And we do prefer finder grids, as Anna showed also. He's showing here the size for comparison, and if you see here, this is basically a lattice, and on it is a carbon film that supports the cells. So we can either grow cells on that lattice, or we can deposit cells. And you see bright field here, just white visible light, and the fluorescence. Before I went into the methods, I wanted to say that we spend a lot of time making sure that the samples we prepare are well prepared. So bad samples will give you back the bad data. That's just universal throughout disciplines. And this is the type of sample we ideally want, where we have quite a few cells. You can see them bunched up. And then we try to avoid the overpopulated grids, which will give you invariably bad data, or underpopulated grids, which might stress the cells. And I'll start with our microscopes. I'll start by showing the cryosim first. This is a super resolution 3D cryo-fluorescence imaging. And our samples are always at this stage in the workflow will have been vitrified. So they go from in solution to rapid freezing. So we preserve all the sensitive structures within cells. We definitely use some mapping facility after that to make sure that the samples are good, and then they can enter the first microscope, which is the cryosim. It's structural illumination. We use, as I said, vitrified samples on grids, and we have 4 lasers, so we can use the full excitation potential within the visible light spectrum. We can do geofluorescence register, and we can look through fairly thick samples up to and sometimes over 12 microns. The resolution is less than 200 nanometers, is entirely dependent on the wavelength of the excitation. This is fully commissioned and available to anybody who needs it. And I'm showing here as an example fluorescent beads, 125 nanometers, and the improvement you get with cryosim, as well as a cell. You clearly can't see the cell fluorescence, but you can see vesicles around the nucleus. This empty space here is the nucleus of the cell. This is a mammalian cell. And these vesicles contain a fluorescent marker. And you can see the improvement from general bright field to super resolution. Again, this is three-dimensional, so matches very well with the soft X-ray tomography that I will describe in a second. The same microscope can be used for cryo-destorm, but we're still commissioning that. So we're still working on convincing fluorophores to blink or deplete in cryogenic conditions, which has proven a little bit challenging. So this is work in progress. All right, so moving on to the next microscope, which is located at the far end of the beamline. This is B24. If you walk in, this is what you will see. Admittedly, it's gotten a lot busier lately. If you turn 180 degrees, you'll see the other side. This is our biological preparation. And the same as Mistral, we take light from a bending magnet. We have a single toroidal mirror that focuses the light onto the sample. We have a monochromator, and our microscope is a standard microscope. It has a condenser and an objective, X-ray adapted, and a detector here. So it's direct detection that we do. All right, so as I said, it's a 3D imaging technique. You've already seen there is some results with Anna's work, and it helps to look at the ultrastructure of vitrified whole cells at neonative states. The benefit of this method is that we use X-rays in a specific wavelength, which means that at that wavelength, carbon-rich biological material absorbs a lot more than oxygen, the oxygen-rich media that surrounds it. Therefore, we get natural absorption. We don't need to stain the cells. So we're as near physiological as possible, bar the fact that we need to vitrify, so we rapidly freeze. We do collect tilt series, so we collect projections of the samples at different angular intervals, and our resolution, depending on the optics, can be 25, 40 nanometers. Of course, with a higher resolution, you sacrifice the field of view, so it's a little bit of a decision, depending on the project, which one you go for. And as I hope to prove, it's complementary to other imaging data. Now, what I'm showing on the side here is some data from my student Kamal Nahas, who's working on understanding herpes virus infections. And what you're looking at is two cells. In fact, there's one nucleus at the top left and another here at the right. And all the little black spots that you see inside the nucleus are actually herpes virus capsids. So we're talking in the region of 1,000 angstroms. And you also can see the effects of the overload of viral production to the ultrastructure of the cell membrane over here. So this is more or less what you can expect to have. As another example, I'm showing you here a mosaic showing some U2S cells that we were working with. And this is the raw data. As I said, we're collecting projections through different tilts. So this is what you're seeing. This is a part of a cell, a nucleus here, and you can see lipid droplets. You can see these little dark spots are gold nanoparticles that we use for positional alignment and a lot of organelles. And then if we process this, going from the top through the cell, you can see the level of information you can acquire with this technique, which is quite impressive, thinking that you're going through the thickest part of the cell, and you can see the organization of all the vesicles and satellite. Okay, so bringing both together. Now this comes somewhere in the in silico work here, keeping in mind that from the very beginning when we prepare the samples, we take a lot of care to prepare the samples that in a way that they can be correlated. And this is the workflow that I mentioned. What I'm going to show you here is what we ended up doing, and I'm using a specific example where we were tracking a reovirus distribution within vesicles in early infection, in early times post-infection, and here's the X-ray tomogram. So effectively, we'll bring the three-dimensional fluorescence information and align it perfectly within the ultrastructure of the cell, which we attain through X-rays. The two microscopes, as I said, X-ray and cryosim are here. So how we go about doing this, and I've mainly focused on the method itself, is we collect the sync data, And then we move, rotate, and align the area that we see in the X-rays. So this area has been moved now, and you can see here red fluorescence, which corresponds to the reporter molecule that we're using, and green fluorescence for the viruses. And here's the X-ray tomography data of that specific area. You can see this is an interface between two cells, a nucleus and another nucleus here, and the ultrastructural organization inside. When we put everything together, all that information becomes a lot more meaningful, because now we can actually assess which vesicles contain the virus and which vesicles have been altered by the virus. We've documented all of this in a recent publication in Cell, and the whole in silico workflow, we've actually put it down. So it's a process that we've charted very well. Okay, so if we look again, zoom in that specific area, and you see that this is the interface between the two cells. You can see all the organelles that I was mentioning in vesicles. And then what I've done is I've superimposed here just the viral signal. So we know very clearly that we found out that these dense areas are actually viral in origin. Now, as I said, the X-ray tomography is three-dimensional, so you can see the same area in 3 dimensions. Again, the information is fairly rich here, but putting together with, which is coming with the fluorescence. Sorry, I'm trying to convince my computer to sort of let me do it. There we go. You can see now that the same tomogram becomes a lot more meaningful. Now, I'm not implying that the fluorescence method that we're using has the same resolution, but because of the good localization between the two signals and the method, the cryo-sim gives really, really clean fluorescence, you can see that the localization is pretty accurate. All right. And finally, B24, again, I mentioned that's us. We're here to get in touch. I've put down some research highlights for people, and the 2 key things that I wanted to flag out is we do have a Twitter presence, so please drop by and say hello. And then before I talk about access, I have to admit there's an awful lot of people I haven't acknowledged. This is very much a collaborative effort between us, between the University of Oxford, between the different technical teams at Diamond, between ARPA also. So there's an awful lot of people that contributed in this. Access routes are fairly well mapped, given the COVID situation. Things can be uncertain at times, but the beamline can be accessed through standard access, which is then peer reviewed, but also through rapid access for feasibility studies. Okay, and that's it for me.